So yesterday po Doc Eugene yung virology natin,
Sorry.
virology.
Yes, Doc. Yes, Doc. Immunology
So for
pala.
today po,
Thank you, thank you.
ah, yes po, immunology po ang itatakal natin today, and this is similar to the structure of the session yesterday. So I have arranged the topics in a way na ano po siya, nagpro-progress. So yung question natin, magi-start po with the basics, and by the end of the slides, by the end of the session, natatakal na natin almost lahat ng important na notes or important na information with regards to this topic. So sa TOS po natin, around 10 questions ang kukunin nila for immunology. Okay, so let's start po with question number 1. Try to answer din po sa mga bahay ninyo. And I'll try to slow down. Yeah, so question number 1, the following statements about immunoglobulins are correct, except A, these are glycoprotein molecules produced by plasma cells. Letter B, immunoglobulins may specifically bind to one or few closely related antigens. Letter C, immunoglobulins produce effector functions even without binding to an antigen. Or D, none of the above. So, ulitin po natin. The following statements about immunoglobulins are correct, except these are glycoprotein molecules produced by plasma cells. They may specifically bind to one or few closely related antigens. Or letter C, immunoglobulins produce effector functions even without binding to an antigen. Or D, none of the above. Yan po. The answer here is letter C. So for immunoglobulins to produce biological functions or to produce effector functions, they have to bind to antigens. So we have what we call immunoglobulin valency, and this refers to the ability of our immunoglobulins to bind to one or more closely related antigens. So effector functions, this include complement fixation, etc. Ayan. So our immunoglobulin, this is divided into 3 regions. Yung nakikita po natin dyan, doon sa pinaka-left, yung 4 na chains, this is the Fab region. And this contains 2 pairs of heavy and light chains. Tapos dito siya, malapit dito sa pinakababa, around the hinge region, we have the constant region. Tapos we have the variable region, yung nasa dulo. The variable region, this is involved with antigen-antibody binding. And we have the constant region dito. Hindi siya nagbabago. Constant nga. Hinge region disconnects the Fab region and the FC region. And FC region, heavy chains lang ang makikita sa kanya. Okay, next. So question number 2. This immunoglobulin has the highest concentration in the blood. So we have IgG, IgM, IgA, or IgE. So this immunoglobulin has the highest concentration in the blood. Is it IgG, IgM, IgA, or IgE? The answer here po is letter A, IgG. So 75% of the serum immunoglobulin is IgG. This is a very versatile immunoglobulin, and this is the smallest among them. So pwede siyang pumunta doon sa extravascular spaces via diapedesis because of its small size. Tapos in certain animal species, IgG din is siya rin yung pinaka-major na immunoglobulin produced from the colostrum, or pinaka-major na immunoglobulin seen in the colostrum. An example of this would be our ruminant species. Yeah. So IgG is secreted by the plasma cells of the spleen, the lymph nodes, and the bone marrow. Question number 3. This immunoglobulin is usually the first responder after a primary insult from an antigen. Question number 3. This immunoglobulin is usually the first responder after a primary insult from an antigen. So you have IgE, IgM, IgGT, or IgA. Answer po dito would be letter B, IgM. IgM is also known as the ancestral antibody for 2 main reasons, because siya po yung unang responder and siya yung unang uh, immunoglobulin na pinoproduce ng fetus. This has the second highest concentration in the serum. Pero due to its large size, this is also known as the largest immunoglobulin. Dahil sa laki niya, limited lang siya doon sa ating extravascular spaces kasi hindi siya makaalis via diapedesis. So as exhibited dito sa ating slide, this has a pentameric structure. So limang parang surfaces connected to each other. Then we have the 10-10-10 chains. So we have 10 heavy chains, 10 light chains, and then 10 antigen binding regions. Okay, next, question number 4. This immunoglobulin triggers the release of inflammatory molecules from mast cells after binding to an antigen. So this immunoglobulin triggers the release of inflammatory molecules from mast cells after binding to an antigen. The answer here is letter D, napakita na. So, IgE, this is mainly involved in inflammation as well as parasitic infections. So it acts mainly as a signaling molecule for our basophils and mast cells. As you may or may not remember, basophils and mast cells facilitate or release histamine. Ayun. So IgE, this has the shortest half-life among the immunoglobulins, and this triggers type 1 hypersensitivity or immediate hypersensitivity reactions. Next, question number 5. This type of sensitivity is mediated by mast cells. Nasabi na natin. Pero ulit lang, this type of sensitivity is mediated by mast cells. So the answer here is letter A, type 1 hypersensitivity. So here shown in the picture is a summary of the process kung paano nagkakaroon ng hypersensitivity reaction. So an allergen is captured by an antigen-presenting cell. Usually this consists of our MHCs or major histocompatibility complex, specifically MH2. And the MH2 will present it to the T helper cells. Then T helper cells will react with B cells, and B cells in turn will release IgE antibodies. These IgE antibodies will signal mast cells, and this will cause mast cell degranulation and therefore histamine release. Doon na natin makikita yung mga clinical signs natin. Question number 6. The following statements about specific immunity is true except A, immune response is antigen dependent. Letter B, it is antigen specific. Letter C, there's an immediate maximal response. Or letter D, exposure leads to immunologic memory. So the following statements about specific immunity is true except the answer here is letter C. There's no immediate or maximal response after specific immunity. So the immune system is further divided or based on several characteristics, may be divided into non-specific or our innate or primitive immune system. Then we have specific immunity. This is also known as adaptive or acquired immunity. So non-specific immunity, this is antigen independent, tapos standard lang yung magiging response dito. Non-specific immunity or non-specific immune system, this consists of barriers doon sa body natin which prevents the entry of our offending or antigens. So, prevent nilang entry. This includes the skin, the mucous membranes, the mucociliary apparatus, etc. Several cells are also members of the innate immunity, and this includes yung neutrophils natin, natural killer cells, etc. Exposure results in no immunologic memory. So, kahit repeated yung exposure natin, same at same yung magiging result. And the purpose lang nito is to try and stop immediately yung mga nakakapasok na antigens. So specific immunity, this is antigen dependent, and mayroon tayong lag time because this allows the body to recognize and prepare an immune response sa mga antigens. This is antigen specific, so repeated exposures will lead to faster immune response or better immune response. Ayan. Tapos meron tayong immunologic memory for our adaptive immune system, or also known as an anamnestic response. Okay, question number 7. Which cell from the innate immune system is important against viral infections and malignancies? Which cell from the innate immune system is important against viral infections and malignancies? So neutrophils, natural killer cells, macrophages, or monocytes? Yeah. Answer here would be letter B. We have our natural killer cells. So they resemble lymphocytes in morphology. However, they are larger and granular. They contain perforins then. So they recognize normal and malignant virus-infected cells, pero non-specific manner as compared to our cell-mediated immunity. Okay, number 8. The following are good characteristics of an antigen except A, stability or stable, letter B, complexity, letter C, foreign, D, none of the above. So the following are good characteristics of an antigen except stable or stability, B, complexity, C, foreign or foreignness, letter D, none of the above. The answer here is letter D. All of these are characteristics of a good antigen. So an antigen is also known as antibody generator, and a good antigen can produce a robust or a good immune response. So if an antigen is not stable, this will not allow the body to recognize and prepare an appropriate anamnestic response. The more complex an antigen is, mas maganda rin yung magiging immune response natin, and usually this would mean na marami silang amino acids na present. And foreignness, it's better if the antigen does not exist in the animal. So as foreign as possible, mas maganda. Question number 9. The following statements describe the FC region of immunoglobulins except letter A involves the activation of the complement system. Letter B binds to the receptors expressed on phagocytes. Letter C composed of constant domains of both heavy and light chains. Letter D, none of the above. Ayan. So the answer here would be letter C. Kasi heavy chains lang po ang present doon sa ating FC region. As opposed doon sa Fab region na nagko-contain ng two pairs of heavy and light chains. And remember po, the FC region, this will bind with our macrophages. However, our Fab region will bind to our antigens. Okay. Question number 10. Acquisition of passive immunity is possible through the following methods except letter A, administration of antibodies. Letter B, administration of pathogens. Letter C, administration of specific lymphocytes, or D, all of the above. So acquisition of passive immunity is possible through the following methods except administration of antibodies, administration of pathogens, administration of specific lymphocytes, or D, none of all of the above. Sorry. Answer po dito would be letter B. So acquisition of immunity may be from two sources or two methods, active immunity and passive immunity. So passive immunity, this is immunity that is acquired from someone else or something else. We have natural and artificial. So natural passive immunity that is passed on from the mother into the young through the use of colostrum, mother's milk natin. Artificial, these are antibodies acquired na binibigay usually ng attending veterinarian or ng clinician. So an example of this, especially for small animal practice, is the administration of Canglobe P or Canglobe D. This contains IgY antibodies. That is an artificial form of passive immunity. For active immunity, this develops after exposure to an infection or after administration of vaccines. Technically, administration of vaccines, especially those that are live attenuated vaccines, is an example of pathogen administration kasi pwede magkasakit yung animal if immunocompromised siya. Pwede tayong magkaroon ng mild fever, etc. Number 11. Question 11. Which of the following is not a primary lymphoid organ? So, which of the following is not a primary lymphoid organ? Is it A, Peyer's patches? Letter B, bursa of Fabricius? Letter C, bone marrow? Or letter D, spleen? So the answer here po is letter D, spleen. So what are primary and secondary lymphoid organs? So primary lymphoid organs, these facilitate the production as well as maturation of our lymphocytes. So for different animal species, iba-iba po yung ating primary lymphoid organs. For example, yung bone marrow, that is a primary lymphoid organ for mammals, especially young mammals. However, once they reach adulthood, bone marrow becomes a secondary lymphoid organ. For birds, primary lymphoid organ specifically producing B cells or B lymphocytes would be the bursa of Fabricius. Yan, for T cells, thymus, no species. Tapos, secondary lymphoid organs, these are important in trapping antigen and doon lang magmamigrate yung ating mga lymphocytes or BNT cells kapag meron na silang nahuli. So spleen is special because it traps blood-borne antigens. And this is divided into two regions, the red pulp and the white pulp. So the red pulp, this traps yung mga dead or dying RBCs natin. And the white pulp, dito nangyayari yung antigen trapping naman niya for our spleen. Ayan. Next. Which of the following statements accurately describe the primary lymphoid organs? So they enlarge as a response to antigens. They are sites in which lymphocytes develop and mature. Letter C. They are sites in which lymphocytes encounter antigens. Or letter D, none of the above. Ayan, the other descriptions here, describe nila yung mga secondary lymphoid organs natin. For example, letter A. For lymph nodes, especially dun sa mga taong nagkakalagnat or sa animals na nagkakaroon ng fever, we'll notice na medyo lumalaki yung kanilang lymph nodes due to the presence of antigens that are trapped by those structures. Ayun, so the answer here would be letter B. Ayan. So primary lymphoid organs, hindi dyan makaka-encounter ng antigens. Lagi siyang sa secondary. Okay, number 13. These are round layered bodies found in the thymic medulla. In certain animal species such as cattle, these bodies contain IgA. So these are round layered bodies found in the thymic medulla. In certain animal species such as cattle, these bodies contain IgA. Is it A, Hassel's corpuscles, letter B, lymphoid follicles, letter C, bursins, or lymphogranular complexes? The answer po here is letter A. So Hassel's corpuscles, these are concentric clusters of thymic epithelial cells, usually found in the medulla of the thymus. So they stimulate thymocyte proliferation by secreting certain growth factors. Ayan. Number 14. So a lymph node is, so this consists of submucosal masses of lymphoid tissue penetrated by radially branching extensions of mucosal glands. Or B, this consists of reticular networks filled with lymphocytes, macrophages, and DCs through which lymphatic sinuses penetrate. Have lymphatic sinuses that contain numerous red blood cells, or a lymph node is a specialized lymphoid organ for blood-borne antigens. Ayan po, answer po is letter B. So ganun din, the main function of the lymph node is to trap and facilitate interactions between T and B cells, specifically helper T cells. So yung DCs na tinatawag natin, those are dendritic cells. And during cases or during instances of infection, they migrate and accumulate in lymph nodes and they will trap the antigen. Doon na, magkakaroon na tayo ng antigen-antibody complex. Number 15, this is related to the question. So number 14. So what is the correct order of events that occur in response to an antigen after they are trapped by dendritic cells in lymph nodes? Yeah. Try to analyze po the question and try to answer po on your own lang muna. I'll explain this in a few seconds po. Okay, the answer po dito is letter C. So here's our flowchart. So when microorganisms invade our tissues, resident dendritic cells migrate into lymph nodes that drain the particular area. For example, yung head or neck region. Excuse me. Then the dendritic cells will form a web to trap these antigens. Tapos, they accumulate in the paracortex and cortex. After the antigens are captured, they are presented to T cells, especially T helper cells around the paracortex. And these T helper cells or helper T cells will interact with B cells and migrate at the edge of the primary follicles ng ating lymph nodes. Tapos B cells will move to the medulla to secrete yung antibodies na necessary for defense. Okay. Next po, number 16. Which animal species have hemal nodes? Number 16, which animal species have hemal nodes? Is it equines, porcines, avians, or bovines? The answer here is letter D. I just want to add pala, mayroon tayong special na immunoglobulin present sa horses that is known as IgGT. Okay, so hemal nodes, also known as hemolymph nodes, are scattered around the small blood vessels of the animal. And yung function nito would resemble a spleen. So this drains blood and filters yung mga bloodborne infections natin. Number 17, which of the following CDs are found on B cells or B lymphocytes and functions for antigen presentation? So which of the following CDs are found on B cells or B lymphocytes and function for antigen presentation? So CDs, these are known as clusters of differentiation. And we have specific clusters of differentiation found on B cells or B lymphocytes. The answer here po is letter C. So this is a summary of CDs found on B cells and T cells. So CDs or clusters of differentiation are used to distinguish lymphocyte lineages. So they have specific functions. These are based on molecules on their membranes. Yun po, si MHC class 2, main function niya is for antigen presentation. Okay. Tapos important naman for T cells, we have CD4 for TH cells and then CD8 for cytotoxic T cells. Next, number 18. The following statements regarding T lymphocytes are not true, except. So the following statements regarding T lymphocytes are false, except they utilize antibody molecules as the receptors. They can utilize free antigens. May typo. D and they can free antigens. They can utilize free antigens. C, they have receptors for antigens on their membranes, or D, none of the above. The answer here is letter C. T cells have antigen molecules on their membranes. They can recognize yung free antigens natin, however, if they are not bound specifically to antigen-presenting cells, hindi nila marirecognize yung mga. So they have receptors for antigens on their membranes, however, T cell receptors yung ginagamit nila, as opposed to sa B cells which utilize antibody molecules. Ayan. Number 19. Which of the following T cells respond to antigen-bound MHC class 2 molecules? So which of the following T cells respond to antigen-bound MHC class 2 molecules? T helper cells, cytotoxic T cells, T suppressor cells, or NK1 T cells? And po. Letter A, T helper cells. Ayun. So T helper cells, after binding with our MHC class 2 molecules, will produce two types of responses. We have TH1 and TH2 response. So TH1 response supports inflammation and activates mainly T cells and macrophages. However, TH2 response activates B cells. Number 20. These are macrophages that are located in the liver. I repeat, these are macrophages that are located in the liver. Is it A, mesangial cells? Letter B, histiocytes. Letter C, microglial cells. Or letter D, Kupffer cells? Sir po dito is letter D, Kupffer cells. So macrophages and monocytes, these are phagocytes po that are found in several places or different places. So depende kung saan sila mahahanap, magpapalit po or magbabago yung kanilang naming scheme. So monocytes, these are found in the circulation, and macrophages are found in tissues. Usually they are free in tissues. When they reach the lung, they are known as alveolar macrophages. Connective tissues, they are known as histiocytes. for liver, Kupffer cells. Letter D, kidney, we have mesangial cells, brain microglial cells, and bone, we have osteoclasts. Ayan. So these macrophages are normally in a resting state. However, kapag nagkaroon tayo ng antigen, doon ma-stimulate yung kanilang activity. Okay. Number 21. Which pathways of the complement system incorporate the use of C5 convertase? Ayan na. So which pathways or pathway of the complement system incorporate the use of C5 convertase? So A, classical pathway. B, alternative pathway. Letter C, classical and alternative pathways. Letter D, membrane attack pathway. Ayan, medyo mahaba-haba itong susunod na itong section na to. The answer here is letter C. We have classical and alternative pathways. Ayan. So C5 convertase is otherwise known as, ito yung mas mahabang pangalan niya, C1-4B2A3B. So both classical and alternative pathways can produce this, albeit through different mechanisms. Yan, i-discuss natin isa-isa, pero sa next slide na lang. Ayan. What is the correct order of events that occur during activation of the classical pathway? Ayan. Nang mahaba-haba. The answer here po is letter C. So C1 activation, followed by C4 and C2 activation, then C3 activation, and finally generation of C5 converters. Ayan. So initially po, kailangan before mag-start yung classical pathway natin, we'll have to rely to an immune complex or an antigen-antibody complex along with calcium. So this binds or this reacts with C1QRS, and this results into autocatalysis of C1R, and C1R cleaves C1S. Ayan. So C1S will react with C4 and C2. So yun po, please remember that C1S has the ability to cleave C4 into C4A and C4B. And it can cleave C2 into C2A and C2B. So dun muna tayo sa C4. So C1S will cleave C4 into C1A and C4B. C4 will remain free. And C4B will bind with C1. Ayun. Then for C2 naman, this will be cleaved into C2A and C2B. C2B will remain free. And C2A will bind with the C1, C4B complex. Yung C1, C4B, C2A complex is also known as the C3 convertase, and now this has the ability to cleave C3 into C3A and C3B. Ayun na. So after cleaving C3 into C3A and C3B, C3A will remain free while C3B will bind with our complex. Yun yung mahaba kanina, and that is known as C5 convertase. And production of C5 convertase will put an end or will mark the end of the classical pathway as well as doon sa alternative pathway, as long as meron na tayong C5 convertase. Now C5 convertase, this has the ability to cleave C5 to C5A and C5B. More on this sa mga susunod na slides natin. I hope nasundan po kasi medyo complicated. Nahirapan din ako dito ng student ako. Okay, so number 23 po. What's the end product of the lytic pathway? Number 23, what is the end product of the lytic pathway or the membrane attack pathway? Pakita ko lang po yung slide ulit. This is the lytic pathway or the membrane attack pathway. So ano po yung end product natin dito? The answer is letter C. Ayun, so the lytic or the membrane attack pathway involves C5 hanggang 9. So C5 is converted into C5A and C5B. C5A will remain free while C5B will bind to C6 and C7. Then makikibind din si C8 and magkakaroon ng insertion of C9 forming the C5B 6, 7, 8, 9 complex, also known as the membrane attack complex. Ito po siya. Question number 25. These are the following are biologic activities of complement except. So the following are biologic activities of complement except A, opsonization, Letter B, lysis of microorganisms. Letter C, virus neutralization. Or D, none of the above. Yan po. Answer is letter D. All of these are functions of our complement pathway. So the complement system takes part in both the specific and non-specific resistance of the body, and this generates several biologic products then. So as seen on the screen, these are functions ng ating complement with specific complements. So opsonization to C3b, lysis of microorganisms via membrane attack complex, yung mahaba pinakita nating C5b hanggang 6 to 9. Chemotaxis via C5a, C3a, and yung incomplete na MAC, removal of immune complexes via C3b, muscle degranulation, venous neutralization via classical pathways. Number 26. The following statements accurately describe cytokines except. So the following statements accurately describe cytokines except they are small secreted proteins which mediate and regulate immunity, inflammation, and hematopoiesis. They utilize tyrosine kinase. Cytokines may increase or decrease the expression of membrane proteins, or cytokine receptors are highly specific and bind only to a single member of a given family. And the answer po is letter D. So cytokines, they are almost similar with our immunoglobulins. They can bind to several receptors. Alright, sorry. The receptors of cytokines are redundant. Pwede silang mag-bind sa ibang cytokines as long as they belong to the same family. Kaya sila tinatawag na redundant. Okay, number 27. The following cytokines are produced by activated macrophages, except tumor necrosis factor, interleukin-1, interleukin-2, or interleukin-10. So the following cytokines are produced by activated macrophages except tumor necrosis factor, interleukin-1, interleukin-2, interleukin-10. Yeah, answer po dito is letter C, interleukin-2. So we have two types of cytokines based on their functions. So we have the mediators and regulators of natural immunity. And we have the mediators and regulators of specific immunity. So members of the first one, or yung members ng ating regulators of natural immunity, all of which are produced by activated macrophages. Yung sa specific immunity natin, iba-iba po yung nagpo-produce sa kanila. For example, we have interleukin-2. This is produced mainly by helper T cells or CD4, and sometimes or in rare occasions by CD8 or cytotoxic T cells. Ayan. Among these classifications, may isa lang po na nagfa-function in both natural immunity and specific immunity, and that is known as the interferon gamma. Paki-take note na lang po, baka lumabas sa board. So interferon gamma can function or can mediate both natural immunity and specific immunity. Number 28. Why is tumor necrosis factor important in acute inflammation and in infections with gram-negative bacteria? So why is tumor necrosis factor important in acute inflammation and in infections with gram-negative bacteria? Letter A, to promote T cell division and to increase production of other cytokines. Letter B, mediates the recruitment of polymorphonuclear leukocytes and monocytes to the site of infection. Letter C, acts as an inhibitor of activated macrophages. Or letter D, stimulates development of Th2 cells from naive CD4 T cells. Ang haba, no? Answer dito is letter B. Ayan. So tumor necrosis factor stimulates endothelial cells to express new adhesion molecules. So after stimulation, these adhesion molecules make the endothelial cells, you can say, sticky for polymorphonuclear neutrophils and monocytes. They also stimulate endothelial cells and macrophages to produce chemokines. Yeah. Fever may also be seen after tumor necrosis factor acts on the hypothalamus. Number 29, which of the following statements regarding interferon gamma is correct? So interferon gamma enhances the microbial function of macrophages. Letter B, interferon gamma stimulates the expression of class 1 and class 2 MHC molecules. Letter C, interferon gamma activates polymorphonuclear leukocytes and cytotoxic T cells. Letter D, all of the above. Answer po is letter D. Lahat po yan ginagawa niya. So interferon gamma has broad functions since nasabi nga natin, this acts both in specific and in innate or natural immunity. So in natural immunity, interferon gamma enhances yung microbicidal function of macrophages. Tapos specific immunity, everything else na. Doon sa mga choices natin kanina. Next, question number 30. Which of the following describes the first set reaction? So which of the following describes the first set reaction? Letter A, a single injection of an antigen into an animal that's never been previously exposed to it will produce no antibody in several days. Or letter B, it is a quick rejection of a graft type that has been transplanted to an animal before, which involves the same donor and the same recipient. Letter C, it is a slow graft rejection that occurs during the first time that the transplant has been attempted. Or letter D, a reaction does not exhibit memory response. So alin po dyan ang first set reaction? Ayan. So clue po, this involves the cell-mediated immunity. The answer po is letter C. So first set response is, or this occurs during grafting, sa grafting siya. Similar po yan sa ating anamnestic response from our humoral immune system. So yung first time na nangyayari siya, the rejection is slow and relatively weak. This could take around a few days. Pero if susubukan niyo ulit siya, nagkaroon tayo ng second reaction, magiging mas mabilis na yung ano natin, magiging mas mabilis na yung rejection niya and magiging mas potent. So this also exhibits memory similar to the humoral immune response. Number 31. The following are functions of the cell-mediated immune response except direct killing by cytotoxic T cells, direct cell killing by natural killer cells and killer cells, phagocytosis and killing of extracellular pathogens, or letter D, graft rejection. Since na-mention na natin kanina yung graft rejection, you can just cross this out sa choices natin. So answer po dito is letter C. Phagocytosis and killing of extracellular pathogens. Because this is a function of our humoral immune response. Cell-mediated immune response is mainly involved in killing intracellular pathogens. So this involves your viruses as well as intracellular bacteria, gaya ng mga blood-borne diseases natin. Ayan. Next, number 32. This is not an animal-related factor that may lead in vaccination failure. So this is not an animal-related factor that may lead in vaccination failure. So letter A, wrong strain or serotype of pathogen. Immunosuppression caused by drugs or infectious agents. Immunodeficient state or genetic influences on the immune system. Ayan po. The answer here is letter A, wrong strain or wrong serotype of pathogen. So hindi ka makakapag-produce ng appropriate na immune response, however, pwede ka makakapag-cause ng fever. Ayun, fever or other clinical signs as well. Pwede ka rin mag-cause ng tissue damage if you use the wrong strain or serotype of pathogen. So hindi ka magkaka-develop ng immunity, however, magre-react yung katawan dito. Ayan. So number 33, why are core vaccines in dogs given at 6 weeks of age? So number 33, why are core vaccines in dogs given at 6 weeks of age? A, because of incubating diseases. Letter B, because of the strain of the vaccine used. Letter C, because of MDA. Or letter D, because the animal is immunocompetent at earlier stages or earlier ages. Yan po. The answer is letter C. So at around that age or earlier, the animal is protected by the mother's colostrum. And the colostrum or colostral antibodies, they start to wane or they start to become weaker at around 6 weeks of age. Kaya yun yung recommended age for vaccinations sa mga dogs na. Number 34, which of the following is not a primary lymphoid organ in mammalian species? Number 34, which of the following is not a primary lymphoid organ in mammalian species? Letter A, thymus. Letter B, bone marrow. Letter C, Peyer's patches. Or letter D, cloacal bursa. Well, very obvious. The answer is letter D, cloacal bursa, because this is a primary lymphoid organ for avian species. Number 35. Which complement pathway is independent of antigen-antibody reaction? So which complement pathway is independent of antigen-antibody reaction? Na-mention natin to earlier. Is it A, classical pathway? Letter B, alternate pathway? Letter C, both A and B? Or D, neither A nor B? Ayan po. The answer po is letter B, alternate pathway. Mas maliit siya dito. Because classical pathway, this requires an immune complex in addition to calcium bago mag-activate yung cascade niya, yung complement cascade. However, for our alternate pathway, this begins only with the activation of C3 via factors B and D as well as magnesium, which is all or which are all present in the normal serum of an animal. Ayan. Number 36. Which of the following inflammatory mediators produce pain and increase vascular permeability? So which of the following inflammatory mediators produces pain and increases vascular permeability? A, histamine. B, serotonin. Letter C, bradykinin. Or letter D, prostaglandin. Yeah. Answer po is letter A or C. A and sorry, letter A or C or letter A and C. Yan po yung mga functions nila. So serotonin or all of them, they may increase vascular permeability as well as vasodilation. However, bradykinin and histamine, these are very prominent since they produce pain. Ayan. So histamine, these are produced by mast cells and basophils. Serotonin, this is produced by platelets and mast cells. Prostaglandin, this is produced via the cyclooxygenase pathway. Cyclooxygenase pathway, that is important in pharmacology. Kasi the basis of the function or the basis of the mode of activity of our NSAIDs is based doon sa cyclooxygenase and lipoxygenase pathway natin. So bradykinin, this is produced from the kinin system. Number 37. Which of the following hypersensitivity reactions is characterized by lysis and phagocytosis of circulating antigens? So number 37. Which of the following hypersensitivity reactions is characterized by lysis and phagocytosis of circulating antigens? Is it type 1, type 2, type 3, or type 4? Yeah. Answer here is letter B, type 2 hypersensitivity reaction, otherwise known as cytotoxic or antibody-dependent cytotoxic hypersensitivity. So type 2 hypersensitivity, this involves the death of cells bearing antibodies that are attached to surface antigens. So antibodies may participate in the destruction of cells either by activating complement, yung kanina, or by the action of cytotoxic T cells. Ayan, so the following are, or yung mga nakikita natin dito sa slide, these are examples kung saan nangyayari ang ating type 2 hypersensitivity. So transfusion or blood transfusion, isa siya. Hemolytic diseases, graft rejection, autoimmune reactions, and hypersensitivity from the coating of erythrocytes or platelets by a drug. Number 38. The composition of colostrum varies with species. In humans, IgA is the predominant antibody, while in horses, swine, and ruminants, the predominant immunoglobulin in colostrum is letter A, IgD, letter B, IgG, letter C, IgE, or letter D, IgN. Simple, nasabi na po natin earlier. This is letter B. Question number 39. What is the substance that is often used in vaccines to prolong the effect of antigen administration that would otherwise be possible? Examples are aluminum hydroxide and certain bacterial components. Is it A, adjuvants, letter B, excipients, letter C, vehicles, or letter D, medium? Yeah. Answer po is letter A, adjuvants. Because excipients, these are used in drug forms to provide stability doon sa gamot. Vehicles, these are used for vaccines. Then, however, hindi ganyan yung magiging function niya. Okay, so, our adjuvants are, or they can enhance the immune response. However, certain adjuvants may also produce several side effects such as fever and inflammation. Number 40, last question before I end this session. This response is the basis for higher antibody production during second exposure to vaccines. Parang throwaway question this. You will encounter certain questions na ganito sa boards din. So don't be don't be shocked later. So A, anamnestic response, letter B, double whammy response, letter C, amniotic response, or letter D, immune response. Letter A po yan. So iwan ko na lang kayo with 4 golden rules for vaccine usage. So yan. So vaccinate only against diseases known to occur in the region. So in the Philippines po, yung core vaccines natin is based doon sa endemic diseases around the area. For example, rabies. In other countries, ang anti-rabies administration nila consists or occurs lang every 3 years, 3 to 5 years, depending on the country. However, sa atin, this is yearly due to the high number of our cases. Use vaccines produced by reputable companies. This is common sense. Vaccinate according to instructions of so follow the route of administration for vaccines. So there are certain vaccines na good lang for IM. However, kapag binigay siya via SC, nagkakaroon ng abscess formation, then pwede pumutok yung surgical site, surgical vaccination site. And do not vaccinate sick animals because down ang kanilang immune response, hindi kayo magkakaroon ng appropriate na antibody production. So that concludes po yung session for today. Thank you for listening. I hope may napulot din po tayo doon sa mga questions natin. Yung manuals po, I'll send them by weekend. Yun po, thank you.
Thank you, Doc.